Hey everyone, it's Jay. Today we're going to be talking about insulin signaling, which we have to understand before we talk about type 2 diabetes mellitus. It's going to be helpful to have reviewed the videos on homeostasis and negative feedback, as well as a video on the normal insulin feedback loop. Those videos are linked in the description and should be linked at the end of this video as well. As a quick review, we need to get to the effector tissue where insulin signaling occurs. After we eat, we know that blood glucose rises. The sensor and control center in this feedback loop are in the pancreas, the pancreatic beta cells. So this change is sensed in the pancreatic beta cell. Show this in the image there. The control center, also in the pancreas, the beta cells respond to the increase in glucose by releasing insulin. At that point, the insulin is going to enter the bloodstream, and it will travel to effector tissue. And the effector tissue includes skeletal muscle, fat, and the liver. We want to keep in mind where this tissue is located. It's not in the pancreas. In response to insulin, the effector tissue lets glucose into the cells. So glucose leaves the blood, enters the cells. This lowers blood glucose and restores homeostasis. All right, so I'm going to focus on a fat cell in the arm. Fat cells are easy to draw, and I want to make sure we're focused in the effector tissue. So if we move to the arm, we can look in the skin and the subcutaneous tissue. The hypodermis, we'll find some fat cells. I'm going to draw a cartoon of one of these cells to move forward. All right, so we have a cartoon of a fat cell. Go ahead and draw the blood supply. Let's have some red blood cells, some glucose molecules that are in the plasma around the blood cells, and the blood is flowing from left to right. Now at rest, glucose that's in the blood can't enter fat cells. Glucose is a polar molecule, hydrophilic. It can't cross the hydrophobic lipid bilayer of the cell membrane. On the other hand, there are glucose transporters that can be inserted into the plasma membrane. And if the glucose transporters are in the plasma membrane, glucose can enter the cell through that transporter. There's the concentration gradient that drives glucose from the bloodstream into the cell, because as soon as glucose enters the cell, it's either broken down for energy or stored as glycogen. Now at rest, there are no glucose transporters in the plasma membrane of this fat cell. The glucose transporters do exist, but they're in vesicles inside the cell. The glucose transporters that are in a fat cell are called GLUT4 transporters, and these are transporters that are responsive to insulin. In addition to the glucose transporters, there's another protein that's present in the fat cell in the plasma membrane, and these are insulin receptors, which I'm drawing here in purple. Insulin receptors are not transporters. So glucose doesn't enter the fat cell through an insulin receptor, but through the glucose transporter. These are separate proteins. So this is what we see in the resting state. In this state, the insulin receptors are present in the plasma membrane. They're not bound to insulin. The glucose transporters are inside the cell, ready to be inserted into the plasma membrane, but they're not there. And glucose can't enter the cell. Now we're going to look at how insulin affects the target tissue. Insulin arrives from the pancreas. Some of it binds to the insulin receptor. At that point, the insulin receptor changes shape. This causes a signal to be sent from the insulin receptor through the cytoplasm. Some of it arrives at the vesicles containing GLUT4. Then those vesicles fuse with the plasma membrane of the effector cells, in this case a fat cell. At that point, glucose can enter the glucose transporters, those GLUT4 transporters that have been inserted into the plasma membrane. So blood glucose falls and homeostasis is restored. So back at our big scheme, restoration of homeostasis, this is what we're after. All right, quick summary. After we eat, blood glucose rises. That increase is sensed in the pancreatic beta cells, causing them to release insulin. Insulin travels to the target tissue, which includes skeletal muscle, fat, and the liver. In skeletal muscle and fat, there's a signal that's going to cause the GLUT4 glucose transporters to be inserted into the plasma membrane of those cells. At that point, the concentration gradient drives glucose from the blood into the cell. This lowers blood glucose and restores homeostasis. If we understand this, we need to watch one more video on feedback inhibition, and we're ready to talk about type 2 diabetes mellitus.